Hello everyone, I'm Dirk van Hyfte. I'm the CTO and co-founder of BioStrand. And at BioStrand, we are passionate about building the next generation technology that makes omics data analysis truly effective. So let's start with looking at the hurdles in current omics data processing. Well, we can conclude there is no common language. There is no common language that glues all together the different data sources, and especially in multi-omics, that contains lots of domain-specific information. There is a ton of bioinformatic tools that needs very specific knowledge to work within, and there is a massive amount of new data generation that even outperforms computer data analysis possibilities. So a substantial amount of knowledge and results remain untapped. And so at BioStrand, we made a discovery. We found very specific signatures, subsequences, that contains very specific information about the structure, the function, the position of sequences at the different omics layers. So they are a kind of intermediate layer between the DNA, RNA, and amino acids. They can be seen as very rich data objects, and in a certain way are a kind of transversal language that glue all these layers together. We have built our BioStrand platform all around these Hyfte patterns. And you could look at our platform from different perspectives. One perspective is at the sequence level, where we see a fully integration of the different omics layers between the DNA, RNA, and amino acids. On the other hand, we add also structural 3D information and even physical-chemical properties to that data. And that data can come from proprietary data, but also public open source data. And especially here, we can use this knowledge to look for protein-protein interactions or binding predictions, epitope predictions, and so on. Another dimension is the unstructured textual information, again coming from public data sources, but also your own proprietary data, for instance, like PubMed, scientific reports, and even electronic patient record systems, so we can tap in that area of data. And that's all combined with formal structured information that also resides in knowledge bases. So what makes our platform so unique? Well, we are using a bottom-up approach, so really from the data itself, and glue together the information that's coming from a sequence to the textual level. So on the one hand, we have our Hyfte integration at the sequence level, again, between the different multi-omics layers. On the other hand, we have our very specific natural language processing technology that can analyze unstructured textual information. And that all in one single platform. So let's a little bit look at the workflow of our platform. Well, we can start off with data coming from human, animals, fungi, viruses, and so on. So really our platform and our Hyfte patterns are species independent, and you can have your own data that is indexed by the Hyfte patterns, but also public data sources together with their metadata. That is indexed by these Hyfte patterns, and also the textual metadata is analyzed by our NLP engine, and it comes all in one single fully integrated model. And the interesting thing is, once you have ordered all that data, then you really can use AI and machine learning techniques to compare, to identify, and even learn and predict certain features that are inside your data. This can help enrich with this NLP analysis to build new hypotheses and test this hypothesis with your data. So let's look at an example. We start off with 1 million sequences of the coronavirus. And of course, all this data contains information, so metadata about the geography, certain variants, epitopes, even clinical outcome data, and so on. We are indexing that data with our Hyfte patterns, and each Hyfte pattern has also associated this metadata labels, and it all resides in one multi-dimensional information model. And again, on this model, we apply our AI machine learning techniques to support the classical AI functions such as clustering, identifying, and predict certain features. What's very interesting of our model is that once it is indexed, you can easily add new incoming data. So you really don't need to rebuild the model from scratch when you have new data. And that helps us also to follow, for instance, the evolution of the virus mutations in the populations and so on. So it's a very easy to contain and adapt model. Let's further look at one single example where we have looked at, is there an intersection between the microbiome and the coronavirus? And as you see, well, there are quite a lot of intersections at the sequence level between the coronavirus and certain microbiome. And let's look at one single further example, and that's an element that resides also in the Mycobacterium tuberculosis. So the bacil that is, let's say, causative for tuberculosis. If we look further at the way we do our NLP analysis, then we have to say that we really start also from the data itself. So really a bottom-up approach. And so what is really important to understand of our NLP technology, that is that without predefined knowledge, so without knowing what's in a certain domain, we can immediately find all the concepts and the relationships between these concepts without predefined knowledge. And that's really key. So it's really in a domain agnostic way, we can analyze this concept patterns and extract that from the data. And then in combination, we are using classical, for instance, transformation models like the BioBird and so on, but also combined with ontologies and dictionary and are doing the matching. And that combination of this bottom-up and top-down approach makes it very powerful to do the further analysis. So let's look a little bit further on how we do this bottom-up approach at the NLP level. Well, let's say you have a sentence, 2 patients are suffering from congestive heart failure. The first thing what we do is relation detection. So we identify the relation between different concepts. And mostly they are verbs, not always, depending on the context of a concept. But once you have detected the relational element, then let's say left and right of that relation, you find your concept. And in this case, we know exactly that's about 2 patients that have congestive heart failure. So we are able to grasp these meaningful word groups again without predefined knowledge. And that helps to understand also the right semantic of the sentences that you can find in this unstructured information. And let me explain this, why it's so important to understand this or to grasp this semantic word boundaries with the following example. Let's suppose you are looking for dopamine. So dopamine is indeed a small molecule, it's even a neurotransmitter in the brain. But there exist also other words that contains the word dopamine. For instance, dopamine receptor. And so it could be a very specific receptor in the brain that is working with this neurotransmitter. On the other hand, you also have the word dopamine receptor antagonist. So that's a chemical drug. It could be even a specific medicine that is working against this dopamine receptor. Another word is, for instance, dopamine receptor gene. could be a molecular sequence, or even a dopamine receptor gene mutation. So as you see, it's really important when you really want to understand in a meaningful way your text that you grasp these meaningful word groups. Let's look at a sentence. It looks maybe a little bit a complicated sentence, as you can see here. So the first step we are doing is detecting the semantic word boundaries. And in this sentence, we can find 5 different word groups. The next step is, for instance, using the famous BioBird or transformer architect models and do the tagging of these concepts. And then you will see that most of these concepts are tagged with the high-level concept gene, so that's fine, that's good. But there are also parts, and you can see it, it's indicated as leftover on the right. So the word interaction and activation is not tagged at all. So it's really not seen, not recognized by this BioBird models. But that means it gives really different interpretation on your sentence. And especially in English, where the last word of your concept can really point to a very specific meaning, as we have seen with the examples of the different dopamine expressions, then here the word interaction and activation really makes sense. And that is also the words that we are mapping that with dictionaries and ontologies, so to have a full coverage of the concepts. And from then on, it's important that you have the capability to do relation extraction or knowledge discovery. So let's go back to our example where we have looked if there is an intersection between the microbiome and the coronavirus. And so we have indexed the whole PubMed abstracts as an example, so the 30 million abstracts, and we are looking for abstracts that contain both word groups like tuberculosis and, for instance, also corona-related terms. And in the different colors you can see, so in green, for instance, all the diseases that are mentioned in that graph, the different organisms, and so on. And indeed, in the proximity of the COVID-19 severity, you can see terms that are relating to Mycobacterium tuberculosis exposure history. So both terms are in the proximity of each other, so they end up in a specific sentence. And of course, there are many kind of this relation between the tuberculosis bacille and coronavirus, but here I've shown one sentence where this co-occurrence of these two terms are mentioned. So that's really looking at the proximity. So concepts that are maybe a little bit farther away, but are in one sentence or even at the document level. But based on our smart indexing principle, we parse all the sentences in concept, relation, concept pattern. So immediately connected with a relation. And in that way, we build another graph representation that represent this direct relations. And also here, of course, we do the tagging at the high level concepts like disease, chemical, and so on, like what's typically done by BioBird. But also here you could then look into the specific relations that are between these specific terms. And here I highlight all the relations that are related to caused by or represent causal relations. But of course, there are a bunch of other relations in this data set. On the other hand, while looking and exploring this kind of knowledge graphs, I also, the word group cytokine storm took my attention. So I also wanted to look further into that concept. And based on our concept relation concept patterns, recognition, you could immediately look at all the concepts and the relations that are immediately connected with cytokine storm, as you can see here on the left side of my screen. And then further, I was also interested to see, okay, COVID-19, that's the second concept relation concept pattern, may be related to cytokine storm. And so what you can do from this concept relation concept pattern, you go immediately to the exact sentences where, for instance, this concept relation concept pattern is mentioned, as you can see with red arrow pointing to that sentence. But then if you look a little bit more into detail to that specific sentence, you see also the yellow markup of that COVID-19 may be related to cytokine storm. And this yellow markup also gives an indication of the certainty of that expression. And in this case, it's may be related to. So it's not that affirmative, not that certain expression. So we also have a way, based on linguistic rules, to do that type of annotation, because that's also important to have a further understanding of a sentence. We are not only looking to certainty of a certain expression, but also negation is also an important markup. And as you see in red, there you have a no specific medicine for this purpose. That's indeed a markup of a negation, also again based on linguistic rules. And even in sentences, we can even expand that negation further down the sentence itself. Apart from this negation, we also have attributes that are related to, for instance, to temporal expressions, dosages, dosage schemas, and so on. So it's a very rich way to understand text. And so if we look a little bit further in the workflow, we really start off with sequences. So in this case, we started with the coronavirus sequence, around 1 million of this sequence. Then in the next part, we look for certain relations between those sequences. And that could be also not only looking for microbiome and for instance, the coronavirus, but it could be also for epitope-paratope interactions or even protein-protein interactions. So there's a way to do that type of analysis. And then based on the retrieved Hyfte patterns with their associated annotations and metadata, we can build a kind of a ranking system, a rank ordering, and that gives us a clue or a very specific annotation of that sequence or subsequences. And that is the starting point to feed in our NLP pipeline to identify these top concepts or the interactions between this top concept in literature. So then really go to knowledge extraction, trying to identify the specific relations that are described or that are used in the literature to describe this interactions between, again, epitope and paratope as an example. And in that way, we really can build a model of doing knowledge extraction, starting from the sequence up to the information that resides in unstructured information. So it's a really powerful workflow. As a kind of summary, well, you can see that indeed we have the integration at the different omics layers. So with these Hyfte patterns and the associated metadata. So these Hyfte patterns are a kind of intermediate layer between DNA, RNA, and amino acids. So it's one single multi-dimensional model. And if we then further look the way we analyze unstructured information, is that it's very important to do the concept detection at the semantic level, so that you really can grasp the meaningful word groups. Because as we have seen, this new transformers tools and so on, they are only partially tagging or doing the annotation, so you are losing the semantic context. So again, that's why it's so important to first identify these semantic word boundaries. Then, of course, it's also important to understand the context around these relational elements. Is the sentence in a part of a negation, with certain uncertainty, or so on. And the smart indexing principle has also the capability to choose what is the level of detail you are looking for. Is it really at the sentence level? Is it around a certain sentence? Is it in a certain part of your document, for instance, only the conclusion, or is it at the whole document level? And the way to add meaning to your NLP tool is, of course, also the combination not only with new upcoming AI tools, but also in combination with dictionaries and ontologies. So I want to thank you very much for your attention. Hopefully also Audrey and Arnoud, you can meet them at our virtual BioStrand booth here at the conference, and hopefully I can see some questions that I can answer. Yeah. Okay, so I see the question is, how do you think the role of omics data in drug discovery will evolve over the next decade? Well, I think how it will evolve is indeed data integration. That becomes very important. I think also the previous speaker has pointed already in that direction. And so you have not only the different levels between DNA, RNA, and amino acids, the combination of metabolics, even 3D structural information, ligand information, ligand binding information, and so on. And so that's why we have built this unique platform around this Hyfte, but that combine this multi-dimensional data representation. And that's, I think, the direction it needs to go. And of course, not only tapping into your, call it, raw sequence data, but also tapping in the knowledge that is in scientific literature and so on. So in the unstructured textual information area, and that combined with formal information that resides in knowledge base. So that's the way I see the direction going on. One other question is, how does this platform compare to its competitors? Well, I think our strong, unique selling point in a certain way is this fully integration. You see quite a lot of players, for instance, also in this NLP area, and they are just focusing in that NLP area. And again, there we can start having discussion on, okay, where they make the difference. But the fact that we, in a domain agnostic way, start building our model and go from there on, so that it's not only just natural language processing, but also more towards natural language understanding. So that's one part. On the other hand, you have players that are working at Omix integration as such, and here, well, we try to combine all these different fields in one multi-dimensional model. So I think that is a very strong point of our proposed technology. Well, another question is what you say is the biggest bottleneck we face in drug discovery. Well, I think one of the bottlenecks is, of course, in the first step, doing the target identification. And One, you start with a certain project, I think it's at that time important to have a full view on all the information that is already available. So having that data integration part solved is an important part. But then once you go further down in the clinical, even drug development process, then seeing as fast as possible, for instance, certain adverse events and so on. And for that, you also need to start looking into the unstructured information that, for instance, resides in electronic patient record systems, and having a way to understand the phenotype that starts popping up when people are using drugs or even in clinical trials. And again, having the combination from the sequence level to the textual level in one model can help to solve that bottleneck. So I want to thank you for your attention, and hopefully again our team will meet you at the conference. Thanks a lot.